Hello everyone. We are going to discuss about the applications of biotechnology in medicine. Introduction. The medical and scientific approaches to human health have two main aspects. They are the curative and the preventive aspects. In the case of genetically determined diseases, prevention is made through genetic screening, antenatal counseling, and gene therapy. In developing countries like India, the development of substances related to prevention of human diseases through the new biotechnological methodology is of great social importance. The methods and findings of recombinant DNA have interfused with the practice of clinical medicine at an amazingly rapid pace, and this has a major impact on the field of health. Antibiotics. The introduction of penicillin and a host of new antibiotics has lifted the scourge of infectious diseases and saved millions of lives. Today, there are about 100 different antibiotics available for use on humans. This is only a small fraction of about 50 mm, and so compounds isolated from microbes which have been shown to kill or disable other microbes. The four major classes of antibiotics are the penicillin, tetracycline, cephalosporin, and erythromycin, which worth over 4 billion in bulk sales each year, and all are superb out of biotechnology. Vaccines. Vaccines are the biological preparations utilized for the prevention of diseases. From the improved techniques like recombinant DNA technology, scientists were able to develop new age vaccines which highly DNA differed from the traditional ones. The immunological insight on vaccination provides us with great deal of information on how the antigenic determinants are involved in obtaining acquired immunity. By using this principle and improved techniques like recombinant DNA technology, scientists were able to develop new age vaccines which highly differed from the traditional ones. New age vaccines. They are of the three types. They include subunit vaccine, peptide vaccine, and edible vaccines. Steroid hormones. Hormones are the body's chemical messengers. They are complex organic molecules with varying chemical composition secreted by ductless glands, which are also known as the endocrine glands. The production of steroid hormones is a combination of chemical and microbiological process. Microbiological modification of molecules in an otherwise chemical reaction is valuable when chemical modification produces two or more isomers. In this, only one of which is active and the other chemical reaction may have a mixture of compounds as the product, whereas the microbial product is only one pure compound. Diagnostic tests. Biotechnological methods allow for the production of highly specific diagnostic tests. In the area of infectious diseases, diagnosis in many cases depends on isolation and identification of specific pathogen, whether bacterium, virus, or parasite. One of the characteristics of being used for the identification of viruses, bacteria, or other agents is their surface structure. The production of monoclonal antibodies by hybridoma technique against specific antigenic determinants of the cellular surface provides methods for the accurate identification of several microorganisms. These monoclonal antibodies are effective in recognizing several parasitic infections such as schistosomiasis, malaria, and trypanosomiasis. They may also be in the identification of some viral infections such as poliomyelitis. Development of organism-specific DNA sequences, which probes would be more sensitive if they could detect suitable specific repetitive DNA sequences from an organism. Short pieces of DNA called probes can be designed to stick very specifically to certain other pieces of DNA. For example, to find out if a patient's blood contains the DNA of a particular bacterium, it provides a diagnostic clue. Not only can all types of microbial infections can be identified, but probes can also indicate genetic defects, detect contamination in donated blood, help in matching organs, and in detecting quality of their seeds. Prenatal diagnosis. Prenatal diagnosis using amniotic fluid is helpful to diagnose diseases caused by recessive genes. The cloned human genes can be used to prenatally diagnose number of genetic diseases. AIDS caused by human immunodeficiency virus, and to diagnose it, many biotechnological procedures have been developed. Gene therapy. Gene therapy aims at replacing a defective gene in an individual, thereby curing that person of their genetic disease. Gene replacement therapy can only be attempted in very few selective genetic diseases. This includes single gene defects in which the molecular details of the affected gene are well understood. Stem cell therapy. Stem cells are creating new waves in personalized medicine. Stem cells are classified into three main classes based upon their capacity to transform cells. They include totipotent, pluripotent, and multipotent cells. The fertilized embryo is an example of totipotent cell due to its ability to develop into an whole organism. The pluripotent, on the other hand, has the capacity to develop into all three lineages, and the best example is the inner cellular mass of the blastocyst, and multipotent cell is highly restricted in terms of differentiation. Stem cells at present are isolated from various parts of the human body. For example, the umbilical cord, Wharton's jelly, that is inner cell mass of the umbilical cord, bone marrow in the menstrual blood of the young female, etc. Stem cells are isolated, purified, and stored for future use in various public and private stem cell banks. Biomarkers. Biomarkers are the proteins that vary in their expression during diseased conditions. Various biomarkers are used for myocardial infarctions such as creatine kinase, lactose dehydrogenase, and troponin C. Pregnancy detection kits are another kind of biomarkers where the human chronic gonadotropin levels in urines are correlated with pregnancy. Cancer biomarkers are another type of biomarkers where the amount of telomerase in the cells is correlated with the diseased conditions. Biomarkers are also used as important targets in the case of targeted drug discovery. Owing to the uniqueness of individuals, there are some problems with identification of biomarkers. Thank you.